right, so our next speaker, Dr. Marc Johnson, is a professor of molecular biology and immunology at the University of Missouri. His work sits at the intersection of virology and environmental surveillance. After 25 years as a molecular virologist focused primarily on HIV replication, he pivoted at the start of the COVID-19 pandemic to wastewater-based surveillance. His lab now uses viral metagenomics on air and water samples to monitor new and emerging pathogens in partnership with public health agencies. And his collaborators have been leaders in characterizing cryptic lineages, which are highly evolved SARS-CoV-2 lineages arising from individuals with long-term infections that are detectable in wastewater, but rarely in clinical samples. Beyond wastewater, his group also collects air and wastewater samples from diverse environments to build a more complete picture of the global virome. And beyond virology and wastewater, Marc is a very keen greenhouse keeper at the University of Missouri and quite well known for that, so you should ask him questions about it. And Marc, it's a pleasure to have you here today.
Hello, yeah. Okay.
Thank you. As they say, now for something completely different. Here's an overview of what I'll tell you about today. Like Nate just mentioned, my background is in virology. This is all very new to me. There will basically be three parts. Shortly, I'll just talk about COVID-19, basically wastewater surveillance. It was a tool we didn't know we really had. We'll go then to actual sequencing from wastewater, something that I didn't really think was going to be practical, but has turned out to be really eye-opening. And then finally, since 2023, we've been working on a project where we do whole wastewater metagenomics as a surveillance tool, and I think there's a lot to be gained from that. So first, how did this all get started? I was in my lab. I was allowed to work in my lab because I was doing COVID work because it was during the shutdown. We get this email from the health department basically saying, is there anyone at the University of Missouri who knows how to do PCR? This was, they wanted to set up a wastewater surveillance program, and their health department did not have the capacity, so they're looking out to other state groups that might know how. And I said, sure, I can help you find someone. I had absolutely no interest in doing it myself, but absolutely no one wanted to do it, and I couldn't say no. So I'm like, okay, I'll give it a try. And I looked at the protocols, and it was taking volumes of liquid, extracting nucleic acid, and then doing stuff with it, which I've been doing for 25 years. So I'm like, okay, I'll give it a shot. Most of us at that point thought this would be over in a few months, but by that summer, this was like a normal weekday for us. We were getting samples shipped to us from across the state, usually arriving in the morning, and we would deliver the results that evening. We were getting upwards of close to 200 samples a week for this. It took time to win over the agencies that this was actually useful, but here's an example of one that we won over quite quickly. As we were monitoring 4 prisons in the state, and we would tell them when they would have cases, and they'd confirm, yeah, we have cases. And then I would say, okay, you just had a blip. They're like, no, we don't have any cases. I'm like, okay. They call back a week later. We just had 40 cases. I'm like, I know. We told you, but we then quickly went from doing 4 prisons to doing every prison in the state. Twice a week, they would have someone from the prison collect the water, drive it to our lab, sometimes 6 hours away, drop it off. We would give them the readout, and then they would know how to act. But despite all this, we saved them probably a ton of money because it was so much cheaper than doing patient surveillance. They then knew when they actually needed to go in and start testing, because that was quite difficult and expensive. Another just point here is one thing early on is when you get COVID numbers, you've got to know what it means. You're reading is the numerator. What's the denominator? And there's always, you need to find a way to figure out how much human contribution there is to wastewater. And we tried a whole bunch of things, but the thing that works the best, hands down, is caffeine and the caffeine metabolites. It's a very accurate indicator of how much human contribution there is to waste. But I just want to make that point because we've been doing this for 4 years. And if you can measure caffeine, you can measure a lot of other things. So the pandemic didn't end in 2020, if you don't remember, and the virus started changing, which was a surprise to me because as a virologist, I'm like, coronaviruses don't mutate. They're very slow to mutate, but that was very naive because everything mutates if there's enough selective pressure. So we started to realize there was these new variants that were called the alpha, which at the time was the UK variant, the South Africa variant, and the Brazil variant. So we were trying to think of how could we very efficiently and quickly monitor our wastewater. And at this point, very little sequencing from wastewater had been done on viruses. So we came up with a strategy where we figured out that if there's going to be mutations in a coronavirus, it will be in the receptor binding domain because this is where 90% of neutralizing antibodies bind. This is where there's the most selective pressure. So we're like, well, let's just build up a system for monitoring that part of the virus. And so we worked out a system. It's very cheap, it's very fast, it's very cost effective. And this is what we've been doing for the last 6 years. And this is sort of a readout of all of the different variants as they've passed through Missouri. When we first started, I said, yeah, I'll just genotype the entire state every week, which I said jokingly, but that's literally what we've been doing. Just a quick look, this is when we first started. We were, I think, the first place in the US to have a dashboard based on wastewater data with the actual lineages. And we got it up just in time for the alpha variant to arrive in Missouri. And then it was really interesting, over the first few weeks, you could see it moving through the state in a very orderly way. It would follow the interstates and then go kind of city to city and then kind of slowly work its way out to the rural parts. And I just want to make this point of how this gives you a better heads up of what's really happening. This is March 23rd readout. This is what the CDC had on Missouri at the same time. They had 5 alpha cases, whereas in fact it was throughout the state already and they didn't know about it. This is probably the first time we really thought the pandemic was almost over. If you recall, alpha sort of was fizzling. It was mid-2021, and then we saw this new variant appear in this, down here, this is a city called Branson. It's a tourist city. A lot of country music goes on there. Close quarters, air-conditioned, possibly singing along, not a mask to be seen. It was a great super spreading event. So we saw this new variant appear in Branson, and it was different. And I looked it up. Oh, that's one of the new India variants. And I would look up. It's like, oh, that's the B.617.2, which is supposed to be the worst of the India variants, which had not really hit the U.S. yet. But it showed up in Branson, and from day one, it was 50%, or it was all of the sample from one of the 2 sewer sheds. So I'm like, that's weird. And then the very next week, this was interesting, it followed a very different pattern than the other viruses, which followed interstates and had very orderly. This one just started popping up in random rural parts of the country, like maybe people who had just taken a trip down to see some country music, I'm not sure. But we had a really big outbreak in, Brookfield, which is, I don't know if any of you recall, but this was ground zero for the beginning of the Delta wave. They had massive amounts of patients in this county. And when we got the sequence, it was entirely this new lineage, which eventually came to be known as Delta. At this point in time, it was still not a CDC variant of concern. It wasn't coined Delta for some time, but we saw very clearly that this was driving a change in patient outcomes. By week 3, it was everywhere. It was basically across the entire state. And it was, in most cases, when we saw it in the sewer shed, the first time we saw it, it was the only lineage we were seeing. So it was really taking over. A good way to look at this is like on the y-axis here is the total amount of COVID from wastewater. In yellow, those are the times when we first saw the UK variant in sites, and you can see UK variant or the alpha variant would arrive and nothing much would happen. But then in red are the weeks when the Delta arrived, and always it's followed within a week or 2 of just massive upsurges. And then a final way to look at it, this was really nice. This was done by a data reporter at Time Magazine, where you could get much more granular data about sequence from wastewater than you could from any other source. So what she took is all of the counties from Missouri and she ordered them on the y-axis where time 0 was the first time we detected delta in the wastewater. And this isn't exactly how it appeared in the magazine. I had her modify it, that she color coded it by vaccine strategy, by vaccine level of the different counties. So at that point in time, there was this new outbreak occurring in Arkansas and Missouri, which was the start of the wave. And we're like, oh, a lot of people at the time were saying, oh, well, yeah, there was not much people getting vaccinated there. But we could see that it really didn't matter of the vaccine level of the different counties. Delta was taking up everywhere. And so this is where got to be kind of useful, because we could tell counties like, Delta arrived, and we could say literally, you're going to start seeing a lot more cases in the next 2 weeks. You need to get ready. And yeah, it really boosted the program. So also in early 2021 is when we had this first weird occurrence that came to be known as cryptic lineages. So this sewer shed right here, Coldwater Creek, if there were a sewer shed that you did not want something weird to happen, it was this one. This is where there was the International Airport. This is where Ferguson is. This is where the Manhattan Project had all of their nuclear waste, which was then buried in a dump, which has, well, they call it a dump fire, like 100 yards away. This is going on now even. So, and the creek, which is where the sewer shed follows, is actually technically a nuclear waste dump. And so you start seeing these weird lineages there, and people kind of go thinking strange things, but we didn't know what happened. So this is just a readout of how we looked at the receptor binding mutations. Up on top are sort of the normal lineages, which we now followed, but it was very weird. So at the time, we had never seen more than 2 or 3 changes in what's called the receptor binding domain of spike. And suddenly we were seeing these SARS-CoV-2 lineages with 7, 8 mutations. And we knew from work by Jesse Bloom and others that these weren't random mutations. These were very clearly antibody escape mutations. And so I was quite nervous that this was like the new thing that was going to take off, but it never did. It actually lasted about 2 months. And then it just fizzled, and we never figured out where it was coming from, but it never left that sewer shed. Shortly after that, I started working with John Dennehy and some others, and we started doing the same type of sequencing in New York City. I was deliberately trying to figure out whether these were particular types of COVID, which were everywhere and they just were under the radar, but they weren't. So the St. Louis variant we never saw anywhere else. In New York City, though, they had their own cryptic lineages, and each one was specific to a sewer shed. And there was a very big red herring, which is that one of the most common mutations we saw was this Q498Y, just letters. But the important thing was this is a mutation that had never been seen in a patient in that period of time. However, it had been seen multiple times when they had taken the virus and they had adapted it to rodents. So I was absolutely convinced that this was probably outbreaks in rodent populations, which would explain why it doesn't spread. They were just in the sewer, right? Makes sense. Totally wrong, but that was my interpretation at the time. So the hypotheses at the time, my first hypothesis was coming from animal shelters, where you have constant influx of new animals, where you could get spread, you could get mutation, whatever. Wild animals, possibly a patient cohort like a nursing home, but the thing I really didn't expect is that it could ever possibly be an individual. Some of the sewer sheds from New York City where we were detecting these had over 1 million people in them, and it just didn't seem possible that we could detect one person that had these, but as you'll find out, that was the explanation. So at this point, though, I started working on an assay because I wanted to figure out what the potential sources could be. So I adapted this assay where you sequence a piece of the 12S mitochondrial RNA, which can just give you a readout of what species are contributing to a wastewater. This is just for fun. This is one of my trial run. This is in my backyard, the Hinkson Creek. I took a sample, just a grab sample, and we took it and we ran the assay, and we found 8 mammals, exactly the ones you'd expect. We found 9 birds, ones that I can confirm all had been nearby, 5 reptiles and amphibians, and about 40 species of fish, all from a 40 mil sample. Yeah, that's pretty cool. So we decided to apply that to our cryptic lineage samples. So starting with one of the sources, it actually made sense. There was, as you expect, a ton of rat. It's actually kind of scary how much ribosomal RNA was, considering they're a lot smaller than us. Pound for pound, you would expect that there was as many rats in the sewer shed as there were people. But anyway, but then other sites where there were cryptic lineages, absolutely nothing, no rat whatsoever. But there's always other animals. There are always cats and dogs and other things. So we didn't really know at this point where it was coming from, but we thought maybe it could be different animals depending upon the lineage. And then Omicron moved in, which was at first, well, I mean, it's terrible for public health. It was also really terrible for our tracking of these because there was so much Omicron that was flooding the wastewater. We couldn't detect them anymore. They just disappeared. And we figured out this cool strategy where we could design something which would only amplify things that were not Omicron. So we could actually go fishing for these cryptics, even though there were at that point a minority of the sequences. And so um, decided to kind of expand it and get samples from as many places as I could. And my colleague John Dennehy said, well, why don't you post about it on Twitter? I wasn't on social media at all at that point, but if any of you have heard of me, it might be because I have since become, I've used social media a lot for science communication, and it's been very gratifying. But yeah, so I posted on Twitter, it went viral, and suddenly I was getting samples from all over the country and all over the world, and we were on the hunt for lots of these cryptic lineages, and we found a lot more. But the first really big one was from my colleague Martin Schaefer in Wisconsin, sent me 8 samples from across Wisconsin. One of them had this flaming hot cryptic lineage. It was just wild, had tons of mutations. And so he said, do you have any more samples, like going back in time, going forward? And he sent us all the samples. It was there every single week at really high levels. So we decided we were going to try and figure out where this is coming from. Again, at this point, we thought it was probably coming from animals, some kind of animal reservoir we hadn't found. This is my colleague David O'Connor's lab going to the dog park, both collecting water and also, excuse me, sir, could I have some of your dog's feces? We had all kinds of different things sent to our lab, but never found the cryptic. And so we decided to track this the hard way using nothing but an auto sampler and a sequencing machine. We started from all of the main lines coming into the treatment plant, and the cryptic was only in one of them. Went to the next pump station. It was only coming in from one of the sites. And then manhole by manhole, they worked their way up until they had narrowed it down to a single manhole. And at that point, we realized it was only had one input, which was from a company. And the health department made contact with the company and said, we think there's someone in your company who's had COVID for a few years and is shedding a lot of virus. Could we contact your company and offer them free testing to maybe find out? And that the company came back and said, yes, but could you first tell us which bathroom it's coming from? They had 2 sets of bathrooms. One was only used by their employees. The other one was also used by the public. And so they wanted to, obviously know whether we tested them. It was only coming from the set of toilets used by their employees. They had about 30. But this was when, I don't know, I should have dawned on me by now, but at this point, we got this sample from downstream of this toilet, and I don't know how many times you flush over a 24-hour period, but I can tell you that this was by far the highest COVID reading I had ever seen. It was 100 times higher than even those prison outbreaks we saw. And I realized that this is coming from a single person, and yes, you could dilute this a millionfold and still detect that they are shedding that much virus. So that's how we figured out that cryptic lineages are actually coming from people. We never figured out who the person was. They were all offered free nasal testing. Most of them took it, but they were all negative. But for reasons I won't get into in this talk, what we believe is happening is in rare individuals, COVID is seeding a GI infection, and this is probably someone who thinks they have IBD, but in fact has a flaming COVID infection in their GI tract. The longest one I know of right now is in Lincoln, Nebraska. They've been infected for over 5 years. Detect them every week. No idea who they are. But in the case of Wisconsin, after they were offered testing, we didn't have a way at that point of doing stool testing to find the person with GI. And then the signal disappeared. No one died, but we suspect that probably whoever the person was realized, either for whatever reason, maybe they were an untreated HIV patient, maybe they were a solid organ donor. But I like to think that they figured out that, oh, this makes sense now and dealt with it, but we'll never know. After 4 months, we finally got our IRB approved for doing stool testing. I actually got to visit the company, and I got to get my selfie with the manhole that we had narrowed it down to, but we'll never probably know what the real end of this story was. But we continue to track cryptics. At times like right now, which I call low tide, when the COVID levels are really low, you start to see them again. I saw 2 new ones last week, one from Arkansas, one from California, each multiple year infections. But just one thing about these. So this is one that I called the New Jersey variant. It was from Newark, New Jersey. It only was around for a few weeks, but it was so divergent. We've seen more divergence in this since this, but at the time it was the most divergent we had seen. So the spike protein has 25 amino acids that actually make contact with the receptor, the ACE2 in this case. This virus had mutations at 15 of 25 amino acid positions. So I reasonably was wondering, had it switched to a different receptor entirely? And luckily, I convinced a colleague, Nicholas Wu at the University of Illinois, to generate this freak Frankenstein spike and test it. And not only did it still bind ACE2, it had the tightest ACE2 binding of any COVID spike he had ever tested. So these are not random changes. These are viruses that are adapting. And they actually give us sort of an evolutionary forecast of where the virus is going to go, because at least in a certain environment, this is telling you what changes are advantageous. Like just to show you, like when these are the cryptic lineages prior to Omicron. These are the mutations we had seen multiple times. I don't know if when Omicron arrived, these were the mutations in the RBD, which was just the wildest thing anyone had seen at that point in time. But of these mutations, all but one of them we'd seen previously, just to show how it really does, it does kind of allow you to forecast what possible changes are for the virus. And just to give you one example of where this did become useful, about 2.5 years ago, if you recall, there was not everyone follows this as much as I do, but there was this new lineage called BA286, which became JN1. At the time, BA286, it was wildly different. It was pretty clearly from a persistent infection that got out, and so it was very different than all of the lineages around. But it was kind of limping along. It wasn't really taking on. And then a bunch of groups, like David Ho and others, had looked at the neutralizing antibody titers, like what is this resistant to? And it still had an Achilles heel. There was a certain class of antibodies we had that it was very sensitive to. But we knew from other work that mutations at 450, one of these two positions would actually alleviate this neutralization capacity. So shortly thereafter, in our wastewater surveillance, this was actually a sequence from Michigan, we saw a new lineage which had a mutation. They're like, oh, well, let's look into that. Looked into our cryptic lineages. Indeed, that was a very favored mutation that was clearly gave fitness and certain advantages. I'm part of this group called wastewater, or called SAVE, which sort of monitors the new lineages, does likes a profiling, helps to decide what happens with vaccines and things. So I wrote to them at the time. At the time, there was only 4 patients in the world that had this lineage, but they were from 4 different countries in Europe, and here we were already seeing it in the US, so it seemed to have legs. We didn't actually find our first US patient until October 17th, like 6 weeks after, but the rest is kind of history. Within 3 months, it had taken off and has been the dominant lineage for the last 2.5 years. But, it's like once you know what's going to be useful, it's useful information, big data. Okay, so this took me to a conference at the end of 22. I got invited to talk about cryptic lineages, in fact, and I met this guy, Jeff Kaufman, who's part of Secure Bio, who's a spinoff from MIT, and they do a lot of surveillance particularly for biothreats. And I had told him that I had done some metagenomics on our wastewater just to see what other things we could monitor. And he's like, can I see that sequence? And I'm like, sure. And I sent it to him, and he wrote back a while later. He's like, I think this could be really useful. If we pay for the sequencing, can you do this every week? And I'm like, okay. That's a lot of data. I mean, doing one of these runs was a big deal for at the time. It was really expensive, but I'm like, okay, yeah, we'll see if this is useful. But this is the thing that, so 30 years ago, I got my PhD where I sequenced a single virus, like 14 kb. That was the biggest part of my thesis. A few years after that, they finished sequencing the Human Genome Project, which at the time to me still seemed like science fiction. It took 14 years and cost around $3 billion. Today, that costs like $200, and you can do it in a day. And the sequence keeps going. The cost of doing sequencing keeps going down. I mean, what we do every week is just utterly science fiction from where I started. It's like I cannot believe we can generate this much data this quickly, but it keeps going down. So I really think untargeted sequencing will be the way of the future and of surveillance, because you can just learn so much. So our procedure is pretty simple. It hasn't even changed. We've played with a lot of different conditions, but this still works, the old viral approach. We start with wastewater, and this is how I see wastewater. It's a bunch of solids and viruses, and we first filter it to get rid of the solids and the bacteria. If you don't do that, all you get is a bunch of bacterial genomes. You then do something to concentrate and remove all the really small stuff, and you're basically left with viruses, ribosomes, and some plasmids. And then we sequence the crap out of it. We go really, really deep. On average, we're going about 1 billion sequence reads per sample. So what do we find in this? 2/3 of this is actually ribosomal RNA, which you can remove it by various methods, but unfortunately, when you remove it, you often pull out things you don't want to pull out. So we just leave it in and just deal with it. And I'm hoping this will become useful, because it actually gives you a lot of information about the microbiome in a temporal and geographic way of like how it changes over time. But of the other, this is kind of maddening. Most of it is unknown. It has no sequence match in the database. We call this dark matter. About 6% is viruses, but it's mostly plants and bacteriophages. About 0.01% is mammal infecting viruses, but when you get 1 billion reads per sample, that's still 100,000 reads, which is plenty to tell you what's going on in that location. So there's kind of 4 levels of the analyses we do. So Secure Bio is most interested in looking for like potentially novel engineered viruses or the next pandemic. Our biggest focus is on public health. I'll talk a little bit about work with doing zoonoses, and also a big part of what we want to do is cataloging the virome. We have 2 dashboards. One is public, anyone can view it right now. We have a second one that we only share with the public health locations that are participating. So just talking about the surveillance, there's a pretty good correlation between what we're detecting in terms of reads per billion, particularly if you normalized it, and what we see in clinics. And that becomes really obvious when you look at things like influenza A. Influenza A, the first flu season, it was present January through March, completely goes away. January through March, completely goes away. I mean, it matches up exactly what we see from patient data. But those are the easy ones, because that's one we're surveilling from. One of the big surprises for us is that the first season, when we were looking at our flus, flu A and B were the minority. The most prevalent flu we were seeing was influenza C, which I wouldn't be surprised if many of you have never heard of it. I hadn't. It's a milder form, but it still infects people, makes them sick. Some people say it. One public health professional said they call it the crud. It's one of those respiratory viruses that you get. It doesn't usually kill you, but well, it was really prevalent the first season, and then we didn't see it at all the second flu season. And then it came back again the third. But since that time, we started off just doing this in Columbia, Missouri, but we have expanded to a lot more sites. We're up to 43 now, but with that, you have more context. So here was our first season in Columbia. Second flu season, flu C wasn't really anywhere. This is our most recent flu season. When it came back everywhere. So flu C was pretty much on par with H1N1 this season in terms of the amount of RNA we see in wastewater, but it's not on any of the panels. So I don't really know whether these are patients that just had a respiratory disease that came back unknown, or whether this is perhaps just completely subclinical, but it's there, it's around, it's just under the radar. Another one that's interesting is intravirus D68. One of the things that just kind of blows my mind is how different respiratory viruses have just completely different seasons. So while flu is, and all of the human coronaviruses are, other than COVID, are January through March, D68 appears regularly in the fall. Another one that's interesting is hepatitis A. It's not there most of the time, but when it appears, it's usually there for a stint, like maybe a month or so. And this is like our sites all over. You'll see there's lots of cases where it will appear, it'll last a while, and then it'll go away. And then there's a whole bunch of cases in California, which were more persistent, particularly in the unhoused population. When we first started, we had never seen measles for like the first 18 months, and now we've seen it about 30 times. And we had this one really interesting case from Chicago that I just want to this still blows my mind. There was a sewer shed where suddenly measles showed up, and we had several reads, and we went to the health department, like, we've detected measles, and they're like, yeah, we have one patient. Like, you sure you don't have more? Like, no, we have one patient. We know the details of it. We know when they arrived. We know what sewer shed they were in, and yes, that was the sewer shed. And so um, Rachel Paretsky, who's does the wastewater for Illinois, decided to dig deeper into this, recover the sequence, find the sequence from the patient, and there was enough polymorphisms we could very clearly say the sequence we detected from metagenomics was exactly the same strain that probably was just that one patient. So this is a sewer shed that has about 300 million gallons a day. So to put that in context, if you took a football field and dug a hole 700 feet deep and filled it up with wastewater, which is a little bit higher than the Washington Monument, that's how much wastewater they get in a day. And they sent me 50 mils. It's just science fiction. It's just unbelievable. I mean, we must have gotten lucky. They must have been a super shower, but still, it's just incredible how sensitive this technique can be. Another one about sort of flying under the radar I like to talk about is the rhinoviruses, common cold viruses. When you look at it like this, of like rhinoviruses A, it just looks like everything everywhere all at once. It's just always present, but it is really so much more intricate than that. So if you take it and you break it apart by all of the serotypes we detect, this was our first spring in Colombia, a whole bunch of different serotypes we detect. Then you go to the next spring, rhinovirus comes back, but it was a totally different list of serotypes. There was a complete shift from season to season. Sometimes there's also a fall wave. The first year there wasn't much of one, but the second year there was. And again, it's really clearly different serotypes with each thing. And what's fun is now when you can look at all of the different sites, this is now overlaying, let's see, I think it's Chicago and Boston and Columbia. The exact same serotypes that we'll see in these waves in Columbia are happening everywhere. So rhinovirus isn't just here all the time, like just a big pool. There are discrete waves of it just happening like nationwide all the time, and we just, sort of don't notice it. It's really, I don't know how this is going to affect public health, but it's just, there's so much more going on. It's so much more intricate than it might seem at first glance. So these are the places we're surveying now. And you might have heard there's some soccer games coming up later this week. And when I started learning about the details of this, I got really excited because I feel like it's a once in a lifetime opportunity because there are going to be people, the teams are from 48 countries. The visitors will be from even more. They're expecting over 5 million visitors, which will stay 1 to 3 weeks on average. The games are distributed across 16 cities. And what the part that really gets me is that it's not random. Each team will play each of their first 3 games in 3 different cities. So there are going to be hordes of people from countries moving in mass from city to city. I'm like, I gotta follow this. We gotta get them. So I've been sort of begging for the last year to enroll with. So we didn't get them all. We got 8 of the 11 US cities are now sending us wastewater at least once a week. Many of them are twice a week. And we have killed ourselves trying to get our turnaround time. So we're down to 5 days between sample collection and our dashboard. So we can go faster, but it gets really more expensive when you start doing it faster because you have to use smaller plates. But that's where we're at right now. Hopefully fast enough to be at least a little useful. And hopefully we can at least be a safety net because we're surveying for a lot of things for World Cup. But I mean, how many of you had hantavirus on your bingo card at the beginning of the year? Or some Ebola strain that we've never heard of? I mean, who knows what's coming? So we just want to be ready for whatever, and hopefully we can contribute. We're also doing, we've started doing various drug testing. So we offered metabolomics for all of these sites. Only a few took us up on it, but we're also monitoring all of these drugs from several of the sites. Okay, now other surprises. So in our first season, we started seeing H5N1. If you didn't know, there was this big cattle outbreak. It started March 25th. It was really expensive. But we actually detected it 2 weeks before it started, and we were 1,000 miles from the epicenter. But as it turned out, there was milk being shipped up to Missouri from Texas, which is probably why we detected it. But still, we were 1,000 miles, and we detected it before it was known. And it took like 4 months for them to figure this out. So we thought, can we use this for monitoring zoonoses? And so we started this project with the NIH-funded SEER network that we started recruiting sites which have animal processing facilities, sort of like if it's going to show up anywhere in the tissues, it will still show up from animal processing facilities. And I went back to my fun species identification, just so that we do species identification on every one of these samples. So if something new shows up, we have a list of candidate species that it could be coming from. Just to show you that different sewage heads have very different composition. This is normal, red is human, mostly human. Chicken processing facility, blue is chicken. And here is one with a hog processing facility where 50% of the ribosomal RNA is actually from pigs. Now, just a little fun with data. We can tell when it's paddlefish season. March and April is when we start seeing these big guys because that's when it's in season. We can see that Atlantis salmon is popular here in Boston, but if you go to Boise, it completely switches and they eat mostly Pacific salmon. We can tell you that Ontario, Canada has an obscene amount of cod, but as it turns out it's because they have the second largest imitation crab processing company in the country, which uses that kind of fish. And so a lot of that ends up in the wastewater. And this is my favorite. Kansas City, sewer should have 500,000 people. Every single week almost every week we detect hippos. The zoo has 2 hippos. Their names are Liberty and Cairo. And there they are every week. But okay, so this actually is useful. So here's one case where it's at least helped us find an outbreak coinciding with when it was found. There was a city called Milan, which has this large pet food processing plant, had a big hit of H5N1. It's a closed system, so it didn't likely come from wild birds. So we looked to our species. What species we find? Well, they had a lot of duck. Like, as it turns out, duck is a very popular protein for dog food. It's hypoallergenic. And so, like, okay, it's probably coming from ducks, and we were ready to tell them, and I decided to check the USDA. Sure enough, the very same week, there was a big outbreak in ducks in Indiana, which is where most ducks in this country are grown. So, and they're not as, they don't drop dead when they get H5N1 the way chickens do. So I'm like, okay, well, that was probably the source. So we didn't actually find the outbreak, but we found it at the same time the USDA did. But it was just to show that we weren't missing things. You really could pick up things that were happening. One of our biggest headaches is anomaly detection. So we've picked up lots of things that are real, that are of public health concern, but we also pick up a lot of things that turned out not to be real. There are a ton of things, particularly like HIV. 99% of the time when you pick up HIV in wastewater is from someone's lentivirus. Harvard has terrible with this. We find all kinds of weird things. We have a pipeline for working this out, but this is something that is really needs to be resolved as this becomes a more broadly used public tool, because we don't want people thinking there's Ebola when it's not there. Here was another example. This is from Palo Alto. There was this parainfluenza virus 5. It doesn't really infect humans. It infects people, or it doesn't infect people. It infects animals. And there was a ridiculously high amount of it in their sewer shed. We could reconstruct the entire genome. We blasted it, and it was a near perfect match to a lab contaminant, something that had been found in tissue culture cells. So what I think is probably there's some company, there's a lot of biotech there, they probably had a reactor where they had cells that were infected with this and just didn't know it. But either way, even if you treat things with bleach, often the nucleic acid still lives, so we detect these things. So finally, layer 4 is the unknown. So most of what we detect has no match in NCBI, but if you assemble it, if you piece it together, you can usually tell it's a virus. And we really focus on the RNA viruses. So this is where we're at. So when you think of metagenomics, I think of it as a picture where you're seeing individual pixels. You can do digital PCR and figure out, okay, oh, that's an elephant, that's a rhino, but it doesn't really tell you much about it except that it's there. There's one of the most common ways of doing this is doing what's called target capture, where you pull out all of the pathogens you care about and then you assemble and you can see, oh yeah, these things are there and this is what they look like. And it's a good approach, but it's more time consuming and more expensive. Our approach is just to sequence everything, and so any animal that we know, we can put the pixels together and like, that's what's there, but still most of it is just a blur because this is sequences we don't recognize. But I want to get to here. Even if we don't care what these viruses are, just being able to say, oh, that sequence is that, that sequence is that, that sequence is that, we can tell the whole picture from it. That way, if something new arrives that we haven't seen before, we'll recognize that it's something new. Right now, we don't know. We have no way of knowing if there's something new because everything is dark matter. So we have a big project with Lawrence Livermore National Lab. I won't go into the details, but one of the fun things is like using just with this information, you can start doing very detailed analyses, and you can tell in an unbiased way exactly what sewer shed any sample came from but just from the viruses present. And this is the sites. This has been a big project of all wastewater data in SRA, the major repository. About half of it at this point came from this project, and that's continuing to increase. But this is the big thing, is that we can do this because sequencing is just cheaper than it's ever been, and we'll continue. So that's my final. A lot of people involved. Happy to take questions.
Great talk. You may already have answered this, but and maybe this is something that you'd have to do calculations on, so forgive me. If you had one patient, one individual with Ebola in Boston, what do you think your positive predictive value would be?
It kind of depends on the state of the patient, but it is definitely within the realm that we would be able to detect them, particularly if they have bleeding in the GI tract.
If they had a GI tract problem.
If they had it, I mean, well, that's what
Yeah, absolutely.
But yes, we wouldn't miss it. Anything that has RNA or DNA genome, we are sequencing. And I don't know the details of how much shedding occurs with Ebola. I don't know if those studies have been done, but yeah. And that's one of the things we're watching for.
But generally the dilution math works out so that you can have enough in the sample.
It could be, it can. Bloodborne pathogens are harder to detect. We don't see hepatitis C hardly at all.
Huh.
But with Ebola, because you're bleeding in all kinds of places, if you are having bleeding that ends up in the stool in particular, yes, there would be no problem. Anything that's enteric is very easy to detect.
Have you ever tried looking at cancer variants like bowel cancer?
There are groups that have considered that. We try to steer clear of the human DNA.
Yeah. Thank you.
Let's alternate sides.
So given the connection between the microbiome and autoimmune disease, which is what I study, is there any chance this could be used for monitoring autoimmune diseases?
I have to believe that of these thousands of genomes that are coming out of our bodies, there will be connections that we just had no way of realizing. There's no way we could have. So yes, I think there's a good chance we will be able to make connections like that. But we're just providing the data. We need AI and various other ways to sort of try and start making those connections.
To the right.
Quite fascinating work. Thank you so much for sharing. I have a couple of questions. The first is, could you comment on what determines what goes into the public database and what is proprietary and only goes to the public health officials that have signed on? The second is, you have many sources of funding listed here, but I'm very curious, how are you currently funding this continuous effort, right? Like someone has to pay for this. Mm-hmm. If you can go into the structure of how that's set up, that would be particularly interesting. And last thing, if there's time, if you can comment on whether or not like these cryptic versions of COVID that you saw, whether or not they have a known target, and whether it's also ACE2 or it's something very bowel specific, and that's why that's necessarily ends up in the bowel. Thank you so much.
So question one, the version that we share with the public, all of the public health groups have agreed to have it be shared public, and we take all of the hot button things on it. We don't actually even list measles or mpox. It's mostly just your run-of-the-mill flu, COVID. The one that goes to public health sees most of the data. They see measles, they see mpox, they see things that aren't supposed to be there, but they're usually annotated with. We think those are real or not. So for funding, there's three main sources of funding. Secure Bio is paying for the actual sequencing, and they get funding through various philanthropies. They are a nonprofit. We have a funder called Inkfish, which funds a lot of surveillance, not just wastewater, which helps us with the personnel and things. And we also have funding through the SEER network, which is NIH funded for the pandemic, zoonotics type stuff. As for the question about the cryptics, So the viruses that SARS-CoV-2 is derived from are largely enteric viruses of bats. And we think that basically these viruses are just returning to that original tropism. And it's actually quite fascinating if you compare. So SARS-CoV-2 is actually has a lot of changes that you would not expect from the enteric viruses. They have a lot of changes where very, very highly conserved regions have changes in them. And in the cryptics, we actually see them very quickly reverting to the enteric sequences at those positions. So we think they're basically just turning back into what their progenitors were, if that answers your question.
One last question.
Okay, let's see. Mark, that was a royal flush. Just connecting Monica's talk with some of actually that other question from Aaron. What if we did something much more long-term, trying to put stuff together about estimated maternal viral exposure versus much more long-term consequences along that kind of autoimmune allergy? Like, how is that going to change outcome in children?
All right.
Children from mothers that had differential viral burdens, and how do we, like, you could do within neighborhood estimates of like long-term consequences of maternal exposures of different viruses. What would that look like?
Surprisingly, most of the viruses are not contained to regions. Those that are contagious very rarely are just within a location. One place where I think that will very likely be useful is when we start doing more metabolomics, where we get readouts of things that are very distinct among sewer sheds that can have long-term consequences.
I guess I would just argue in our social network now that we have a much more distributed exposure to many, many different viruses, and we way underestimate that now versus, let's say, 20, 30 years ago. Something like that. Is that?
Sorry.
Just the way that we're spreading viruses around, and then the exposures of people, including pregnant mothers, is just at a different scale than.
You think the scale has changed, or you think we're just detecting it more now?
I think that we have, with daycare centers and particularly in economically, that there's much higher spread, and we should. I don't know. Is there support for old sewer water versus new sewer water?
God, I wish I had old sewer sedge samples for so many reasons. One thing, though, I'd really like to have, though, is samples that we know are only adults or only children, to sort of figure out. There's a lot of viruses that are so ridiculously widely present, like AACHI, which is a type of, it's one of the most prevalent we see, and it's not really on anyone's radar. I don't know if it's all coming from kids. I mean, if you look at the literature, it's only coming from like outbreaks from eating raw oysters, and I don't think that's happening in Colombia every week. But I'd really like to sort of get a breakdown of what different parts of society are having these different infections, because I know the viruses are coming from somewhere. They're there. Time will tell.
Thank you, Marc, for this really stimulating conversation. And I think this audience is going to think very differently today when they sit on the loo. So thank you all for listening in person and virtually. We are taking a short break before continuing with the program.